So this is one of the first pictures ever taken of the interior of the second largest island in the world, Papua New Guinea. For generations, this part of the world was inaccessible to outsiders due to steep mountains, dense jungles, and the fierce people that lived within it. Over 700 languages are spoken in Papua New Guinea, and the territories that people live in are defined by these languages. And these territories are also fiercely defended by the people that live within it. And one of these people live in the eastern highland regions of New Guinea, and they're called the Four People. These people first encountered Westerners in the 1930s, and these Westerners that encountered the people found people that were living in subsistence agriculture conditions, slash and burn, gathering food from the jungle that surrounded them. They had unique cultural practices, but they also had a unique disease, kuru, that we just heard about in this brief clip that you just listened to. This was a terrifying disease. It was uniformly fatal. It had some really scary characteristics that you saw briefly in the movie clip, and was widespread within these people. By the 1950s, it was the people that knew about this disease, kuru, were worried that it would potentially exterminate the Four People. In fact, at the end of this epidemic, 1,000 of the 12,000 Four People had died from this disease. So in the 50s, a number of scientists were sent by Western countries, including this man here, Carlton Gajdusek, who was a physician from the U.S., to go study the Four People and understand a little bit about the disease so they could try to figure out what was causing it and hopefully stop this epidemic from continuing. So these physicians first wanted to understand some of the natural history of this disease. And so what they did is they studied the people that had it and learned how the disease progressed. And what they found is the disease started with a slight shaking, tremors that the people experienced, and they called this kuru, the people. That meant shivering to them and shaking. That was the first symptom. They then progressed to being able to not being able to walk very well. So these women here exhibit that. They needed sticks to help them walk. They had abnormal gait, ataxia, kind of a shaking walking status. And then eventually they couldn't walk. They even needed help standing, like this woman here. And eventually they couldn't stand. They could only sit, like these children here. And eventually they couldn't even sit. They could only lay. And at that point they would die, inevitably die shortly thereafter. So they had observed what this disease looked like, and the symptoms suggested a neurologic disease. So brain specimens were taken from some of the people that had passed away, and they brought them to their laboratories back in the U.S., and here's what they saw. Here's a section of the brain, and they looked at the section under a microscope, and what you can see these little white spots, these white spots in the tissue. That shouldn't be there in your brain. Those are holes in your brain. They called this spongiform encephalopathy, basically a sponge, a pathology of the brain. Here is a Purkinje cell, this cell right here. This is a cell found in the cerebellum. This is the cell that helps you with balance and walking. So when you have an ataxic gait like these patients had, this is why. These Purkinje cells also had holes in them. They didn't function properly. And when they looked further under microscopes, they saw these brown splotches and these blue splotches and these green splotches. These were plaques that were present in the brain. They shouldn't be there. And when they looked at them under transmission electron microscopy, as shown here, they could see these fibers that were forming in these plaques. And they hypothesized that these fibers were what were actually killing the neurons and causing the disease that they were seeing. In fact, they were proven to be correct in that. So they knew now something about the disease, how it progressed, that it was a disease of the brain. But now they needed to understand why. So where was it coming from? Why was there an epidemic? What was happening with them? And so what they did was they studied the people again to see what did they have in common, those that had this disease, that were different from those that did not. And if you look at this slide carefully, you yourself could figure out what they had in common. They were all the same. It was the women of the tribe that got this disease. It was the young girls of the tribe that got the disease, and young boys that acquired the disease under the age of 6, but not the men. There was something that predisposed these people to the disease, but not the men. And so they started studying some of their practices. Was it sorcery, like the people thought it was? Was it something that they ate? Was it something that they didn't eat? Was it genetics? Or was it an infectious disease? And so they started looking at also some of everything that they ate. They studied all their attributes of these people, and they studied their cultural practices. And they had one cultural practice that was very unique to them, that stood out to them when they found out about it. They were endo-cannibals. They ate themselves. They ate their relatives when their relatives died. It was a funerary practice. And it turns out only the women and the girls and the young boys ate these relatives. The men did not. And that linked these people with something very special, very unique about them. They were the ones that were eating their relatives. They would take the brain, they would take the tissue, they would take the entrails, they would take the bone marrow, they would grind up the bones, they would eat every part of their ancestors, and it was part of their practice and their funeral rites. So now they had a hypothesis, and Dr. Gradishek took this hypothesis and brought it back to the U.S. to test it. And what he did is he took brains from individuals. You can't read their names, but it's important to know their names. These were individuals that died of Kuru. And he took their brains and he brought them back to the NIH laboratories in Maryland, and he injected a number of different monkeys, but specifically injected chimpanzees, our closest related evolutionary relatives. And he injected brain pieces from Kuru into the brains of these chimpanzees and then waited and waited and waited. Now, this disease takes years to develop, and we didn't have a lot of time, but he had to wait, and sure enough, it turned out his hypothesis was correct. And 2 years later, these chimpanzees began to develop the symptoms and signs of Kuru, and when they removed the brains of the chimpanzees, they'd see these same plaques and these same fibers. Indeed, had proven that this was an infectious disease and was caused by ingesting the tissue, the brain tissue, of people that had died from it. And for this work, he won the Nobel Prize in Medicine and Physiology in 1966. Now, shortly after he discovered the cause of Kuru, the government of Papua New Guinea outlawed the funerary practice of eating your ancestors. And this disease takes many years to manifest itself, and so it took time, but over time we saw over many years that the number of Kuru patients declined and declined and declined. But frighteningly, some developed Kuru 50 years after they had eaten their ancestors in the early 2000s. And this is frightening because there was something different about these people than these people. And it turns out they are genetically a bit different from each other. And we're going to come back to that in just a few minutes. This is another man who won a Nobel Prize studying Kuru and related diseases. His name is Stanley Prusiner. And he discovered that this disease is not caused by a bacteria, it's not caused by a virus, it's not caused by a protozoan or a fungi. It's caused by something that he coined a prion, a proteinaceous infectious particle. He hypothesized that this disease is caused by proteins that replicate themselves. Now, this is heretical to biologists. All protein comes from DNA, which then leads to RNA, which then leads to protein. That's how you make protein. You want to make more protein, you start to make it that way. You don't replicate protein. It doesn't work that way. But he hypothesized that indeed it did work that way, and he was proven to be right. And for that, he won the Nobel Prize in Medicine in 1997. So how does this work? Well, it turns out all of us have this very dangerous time bomb in all of our cells in the body, but especially in our brain. It's called a prion protein. We all house it. We all have it. We don't actually know a lot about what it does. In fact, you knock it out of a mouse, the mouse is fine. But it has some function we don't exactly know, but it is a time bomb. And so what might happen, and he hypothesized, is that if you get the wrong copy, this Kuru copy of this protein in the brain, it's going to replicate itself. And that's exactly what happens. And so this little movie that just came out in August, from a tour de force of mathematicians and particle physicists developed this incredible movie, showed how this works. Basically what happens, whoops, sorry. See if I can get this to play. There we go. So this blue is the normal protein. It's a alpha helix, and it encounters the mutant protein, which is this beta sheet. And this beta sheet grabs onto the alpha helix and starts to unwind it and refold it and turn it into an exact copy of itself, which is not what you want in your brain. And now you have 2 copies of this bad protein. And these 2 copies can go find 2 more copies of normal protein, and they can convert it to the bad protein. And on it goes exponentially until you develop millions upon millions upon millions of copies of these aberrant proteins. And they can form these incredible highly ordered structures that turn into the fibers that kill the neurons. That's how this disease is caused. It's a prion disease caused by replicating proteins. Now, Kuru is gone. We don't have to worry about it. We don't have to think about prions anymore. Oh, but wait, you really do need to think about prions. Because in the late 1980s and early 1990s, this horrible disease appeared. This is called mad cow disease, or bovine spongiform encephalitis. It is exactly Kuru in cattle. And so this is what the farmers were seeing in the UK. They were seeing cows, more and more and more of these cows that exhibited the signs of Kuru. They were shaking. They had a funny stance. They had ataxic gait. They startled very, very easily. They had staring of the eyes. They made abnormal sounds. And some of these things are shown in these videos of these cattle that were afflicted with this particular encephalitis. And so when they looked at the brains, indeed they had spongiform encephalopathy. These cattle had a prion disease. And you can see that over time in the 1980s, the late 1980s, this is in thousands in this gray, this is how many cattle were actually diagnosed with this disease in the UK. And you can see it was going up exponentially in the late 80s and early 90s. And by the time that this was recognized, then the UK agriculture industry was under great stress. Nobody was willing to buy these cows from the UK. In 1990, this idiot agriculture minister Goomer brought his daughter Cordelia in front of the cameras and the press and said, "See, this meat is fine. We're going to eat hamburgers right in front of you. Cordelia, go ahead and eat your hamburger," which she did. But he was wrong. This meat was not fine. This meat was contaminated with a prion that can cross the species barrier. At the time, it wasn't thought that this could happen. But indeed, for BSE, this does happen. And in the mid-1990s, the first variant Creutzfeldt-Jakob disease cases, basically Kuru, started to appear in the UK and then in Europe, and even a couple cases in the US from people that had eaten meat from contaminated meat. Once this linkage was made, all these animals had been slaughtered, and the disease started to wane. But what we're worried about, this was only in the 1990s. Remember the Kuru, the people that took 50 years to develop the disease? There are people with genetic variations that likely will develop variant Creutzfeldt-Jakob disease that ate those hamburgers maybe 40 years from now. That's what we're worried about. Now, how did this big epidemic occur, you might ask? Why all of a sudden did it appear? And why did it expand so much? That was because of the practices that were in agriculture that were conducted in the UK at that time and in the US, exactly the same. They've made the cows into cannibals. The cows were eating themselves. When their colleague died of bovine spongiform encephalopathy, nobody wanted to eat that downer cow, so they would take that cow, they'd convert it into protein and feed it back to the cows. So one cow developed this disease. It was fed to a bunch of other cows. They developed the disease. They were fed to more cows. And next thing you know, you had this exponential growth. If you take home one lesson here, don't feed animals their kin. This is not a good idea. It's a great way to cause disease. So now we know about BSE, we can control that. It's an epidemic that's under control. We don't have to worry about prion diseases anymore, right? Wrong. Chronic wasting disease is a disease, it's a spongiform encephalopathy. It's a prion disease of cervids, deer. White-tailed deer, mule deer, elk, moose, all of them are highly susceptible to this particular prion disease. Here's a deer that's wasting away. The exact same characteristics that you saw in Kuru, now occurring in deer. And where did this come from? It came from a deer farm in Colorado in the 1960s. That was where it was first discovered. And quickly it was found to have left that deer farm and was infecting wild cervids in Colorado. And now you can see the result all across the West. This is an endemic disease. Up to 25% of the cervids in certain areas in western part of the U.S. are infected with chronic wasting disease. Now you see 24 states and 2 other countries have this disease as well. And how did they get it? They got it through farming practices. These sick animals were then brought to other farms, people that were raising deer on farms, and then brought that sickness to the wildlife that was adjacent to them. Here's one example. 2 years ago, a farm around Brainerd had chronic wasting disease deer found in it. And a year later, we found that one wild deer was found in the adjacent area. This year, this particular part of the state is open season on deer. There are no limits. You can kill as many deer as you want. They're trying to get rid of the disease. How do you get rid of chronic wasting disease? Outlaw deer farms. We do not need more deer, right? I think we can all agree with that. There's no reason to have a deer farm. We need to outlaw deer farms. The DNR in Minnesota wants to outlaw deer farms. The Department of Agriculture, not so fast about that. But I think this is the right way to do it. And the other way to slow this particular chronic wasting disease, intensive but ethical hunting practices. Take deer. Try to eliminate the overpopulation of deer in our state and in the country. We have way too many deer, much more than naturally our ecosystems can handle. And if we don't do something, this is what we're going to be left with. Thank you.